Roundup was created by the Monsanto Corporation in 1974 and is one of the most widely used herbicides worldwide. This video documents an investigation carried out by the students of GMIT on the effect of Roundup on the aquatic invertebrate Gammarus. We begin the investigation with our student pouring Roundup into a beaker for dilution. This is carried out in a fume hood to prevent inhaling the fumes. The volumetric flasks to be used in the dilution process are pre-labeled by the student. Distilled water is used in the dilution process to minimize any other possible contaminants. The required volume of Roundup is measured out in a graduated cylinder by reading from the bottom of the meniscus at eye level. The herbicide is diluted by measuring out the pre-calculated volume of the Roundup in a graduated cylinder. Pouring this into a 100 milliliter volumetric flask and then adding distilled water until the volume has reached the 100 milliliter mark when read at eye level from the bottom of the meniscus of the liquid. To perform the 1% and 0.5% dilutions, the herbicide is added from the volumetric flask containing the 20% herbicide solution. This is then diluted following the same procedure as before. The petri dishes into which the Gammarus will be exposed to the herbicide are now labeled by a student. Ten Gammarus for each of the 6 petri dishes are caught using spoons and small nets. They are then placed directly into their respective petri dishes. The diluted herbicide solutions are added by a student to the petri dishes, which contain 10 live Gammarus each. After the first 10 minutes, the Gammarus which have died are counted. This is repeated every 10 minutes until at least half are dead. "24 hours later." The remaining alive Gammarus are counted 24 hours after the initial addition of the herbicide. Here we see that after 24 hours 100% mortality occurred in all concentrations of herbicide. However, all specimens in the controlled solution had survived. The LT50 graph plots the time in minutes against percent mortality. This allows us to calculate that a 20% herbicide concentration results in 50% mortality after 12 minutes of exposure. The 24LT50 graph is used to calculate at what concentration the herbicide results in 50% mortality after 24 hours. This cannot be calculated in this case, as all concentrations tested resulted in 100% mortality within this time frame. All specimens were dead after 24 hours of exposure to the varying degrees of herbicide concentration. This indicates Roundup has a toxic effect on Gammarus. The fact that all concentrations had this effect shows glyphosate is a strong toxin. Further testing may be required with larger samples to show this is the case. So, the implications. Glyphosate causes death in Gammarus and is therefore possibly toxic to other species of macroinvertebrate. Without detritivores, far more anoxic decomposition will occur within the environment. The decaying Gammarus will also amplify the ammonia produced by anoxic decomposition. Ammonia will then toxify the environment, and nitrates produced will possibly result in harmful algal blooms. It can therefore be recommended that Roundup not be used near waterways unless with use of a buffer zone. Roundup should not be disposed of into waterways. It is also recommended that Roundup is not used near agricultural drainage ditches, as this may result in Roundup entering downstream waterways. I hope you have found this investigation and its results both eye-opening and stimulating. I thank you for your time.